Hey everybody, welcome back to Curious Things Studio. Today we're going to be learning about meiosis. Have you ever noticed that you and your siblings don't look exactly the same even though you have the same parents? That difference isn't random. It happens because of meiosis. In this video, I'll explain how meiosis works, why it's important, and how it creates a unique combination of traits that make each person different. Okay, now what is meiosis? Well, meiosis is a special type of cell division that produces sex cells that are also known as gametes. In humans, these are sperm cells and egg cells. The main purpose of meiosis is to reduce the number of chromosomes by half and create genetic diversity. Chromosomes are structures inside cells that carry DNA. They also contain the instructions to how we grow and how we look. Most human body cells are diploid cells, meaning they have two sets of chromosomes. So that means 46 chromosomes in total. Sex cells are haploid cells, meaning that they have one set of chromosomes, so they have 23 chromosomes in total. To remember haploid, I think of half. One chromosome in each pair comes from each parent. This is the process that makes these haploid sex cells. Now let's get to the actual meiosis process. So meiosis happens in two stages, meiosis I and meiosis II. One starting cell divides twice and produces 4 cells total. So if you were here with me in our last video, we talked about mitosis. Mitosis used the process PMAT. So does meiosis. So that's less memorization. Anyways, let's get started. So meiosis I is all about creating differences. First is prophase I. This is where the matching chromosomes pair up, and they are called homologous chromosomes. They exchange pieces of DNA in a process called crossing over. This is to increase genetic variation. Next is metaphase I. Chromosome pairs line up in the middle of the cell. Their arrangement is random, so it adds even more genetic variation. Next is Anaphase I. Homologous chromosomes separate. Each side gets one chromosome from each pair. Next is Telophase I and cytokinesis. The cell splits into 2 cells. Each cell has 23 chromosomes, but each chromosome is still duplicated. Next is Meiosis II, which is really similar to mitosis. Meiosis II is similar to the regular cell division, which is called mitosis, which we reviewed in our last video. So prophase II starts and the chromosomes prepare to separate. Then in metaphase II, chromosomes line up in the center. And then anaphase II, sister chromatids separate. Telophase II and cytokinesis, the cells divide again. If you want a more detailed explanation, go watch the first video that we posted. As for the final result, at the end of meiosis, there are 4 daughter cells. Each cell is a haploid, which means that it contains 23 chromosomes. All cells are genetically unique, that means they're all different, and the cells become sperm and egg cells. Now you might be wondering, why is meiosis important? Meiosis is important because it makes sexual reproduction possible, it creates genetic diversity, and it explains why siblings look different from each other. But sometimes, meiosis doesn't go perfectly. A mistake called non-disjunction happens when chromosomes do not separate correctly during mitosis. This can cause cells to have too many or too few chromosomes, which may lead to genetic conditions. Now I'm going to talk about some of the extra information that you might need to know for your class. One thing you might need to know is that unique combinations created by meiosis can lead to incomplete dominance, which means that the traits blend, or codominance, which means both traits show up. And these combinations can also be predicted with Punnett squares. And some traits called polygenic traits are controlled by multiple genes, making people even more unique. So in conclusion, meiosis is the process where one cell divides twice to create 4 unique haploid cells with half the number of chromosomes. Okay, thank you for watching, and see you all next time. Goodbye!